students, and welcome to the video solutions of ANTS FT6, the Botany section. Now, question number 101. The famous botanical garden of Kew is located in. So, the Royal Botanical Garden of Kew, that is located in England. So, going by that, question number 101, the correct answer should be option B. Question number 102. Choose incorrect statements. So, statement A, chrysophytes includes diatoms and desmids. This particular statement is correct. Chrysophytes are found only in fresh water. So we know that chrysophytes are aquatic in nature and they are found in both freshwater and marine water conditions. But in this particular statement, they have mentioned only in freshwater. तो only लिखने की वजह से क्या हो रहा है? ये particular statement गलत हो रहा है. So that is why incorrect statement हमसे पूछा है. Statement B. Question number 102, the correct answer should be option B. Now, question number 103, read the following statements and select the correct option. तो यहाँ पे statement A बोला गया है that majority of pteridophytes are heterosporous. Now, even though we do have pteridophytes who are heterosporous in nature, majority of pteridophytes heterosporous नहीं होते हैं. Among the entire group, there are few limited examples of heterospory. So going by that, it seems statement A यहाँ पे incorrect हो रहा है. So statement A is incorrect. Statement B, in equisetum, sporophylls form distinct compact structure called cone. So equisetum is a member of pteridophytes, and there the sporophylls, they tend to aggregate and result in the formation of a structure called strobilus or cone. So statement B seems to be correct. So going by that, only statement B is correct. So question number 103, the correct answer is option B. Now question number 104 is a match the following question, जिसमें life cycles को उनके respective plant groups के साथ हमें mix and match करना है. तो haplontic life cycle is observed mostly in case of algae. तो majority of algae, they tend to show haplontic life cycle. Then moving on to diplontic life cycle. Diplontic life cycle is observed in case of higher plants. तो उसमें दो include हो रहे हैं. We will have gymnosperms and angiosperms. While haplodiplontic जो life cycle हो रहा है, haplodiplontic life cycle, this is observed in case of two groups, bryophytes and pteridophytes. ये हमारे तीन respective life cycles हो रहे हैं यहाँ पे. So going by that, when we match it, haplontic life cycle A, this is matching up with Chlamydomonas. Algae में यहाँ पे specifically Chlamydomonas का example आ रहा है. So A2. Diplontic life cycle would be gymnosperms and angiosperms, which was 1. So B1. Haplodiplontic life cycle, that is bryophytes and pteridophytes. So A2, B1, C3. So the correct answer for question number 104 should be option C. Now question number 105. Given figures A and B represents respectively. इसमें figure A is for an aestivation whereas B is for a placentation. Figure A, that is representing twisted aestivation, whereas figure B is representing free central placentation. So option B. Question number 105, the correct answer is option B. Now question number 106, which of the following statements is correct? तो इसमें हमसे correct statement पूछा जा रहा है. Pneumatophore is found in Rhizophora. So Rhizophora, a type of mangrove which is found in saline marshy conditions. So they have an advanced type of a root modification. This particular root modification is called to be pneumatophore, which is helping them to perform easy gaseous exchange. The statement A seems to be correct. Axillary bud is found at base of leaflet. Axillary bud leaf के base पे मिलता है, leaflet के base में नहीं. Reticulate venation is found in most of the monocot leaves. This is incorrect because reticulate venation is a characteristic of dicot leaves. In cymose inflorescence, the main axis continues to grow. Cymose inflorescence का जो सबसे characteristic property है वो ये है कि उनका जो main axis है एक point पे जाके उसका growth terminate हो जाता है because of the formation of a flower. So it seems out of all of them जो correct statement है वो सबसे पहला वाला हमारा statement है. So question number 106, the correct answer is option A. Now question number 107, During secondary growth, vascular cambium in dicot stem, the cambial ring in case of a dicot stem, what it does is, let's assume कि ये जो ring यहाँ पे आ रहा है, this is the cambial ring. So what this cambial ring is going to do is, it's going to develop secondary tissues on both sides. Cambial ring का जो बाहर वाला area हो रहा है, उसमें वो deposition कराएगा a layer of secondary phloem. On the other hand, इसी cambial ring का जो inner side होगा, so towards the inner side, it is going to deposit a layer of secondary xylem. So cambial ring, towards the inner side we have secondary xylem, towards the outer side we have secondary phloem. That's the first point. The second point we have to keep in mind is that out of the two secondary tissues, जिस tissue के deposition का rate, जिसके deposition का amount ज़्यादा होगा, that is going to be secondary xylem. It's deposited at a much faster rate and it is also going to be deposited at a much higher amount. So going by that, now when we look at our options, option A, more active on the inner side, forming more amount of secondary xylem. The first glance में क्या हो रहा है? That statement is fulfilling whatever points we just made here. So going by that, question number 107, the correct answer should be option A. Now question number 108, the assertion Lycopodium and Selaginella are heterosporous. So इस particular assertion में problem ये हो रहा है that even though Selaginella is heterosporous, Lycopodium is not. So going by that, it seems assertion is false. Now, reason वाला जो statement हो रहा है, in heterosporous condition, two kinds of spores are produced. So hetero का मतलब क्या हो रहा है? Multiple, more than one. So going by that, the presence of two kinds of spores, that is the possibility of a heterosporous condition. तो ये statement यहाँ पे correct हो रहा है. So reason seems to be true. So going by that, if assertion is false but reason is true, ये वाला statement correct है. So question number 108, the correct answer should be option D. Question number 109. The quasi-fluid nature of lipid enable lateral movement of proteins within the cell membrane. So ये जो particular statement है, this is coming from the fluid mosaic model proposed by Nicholson and Singer. So according to that model, what we know is this, that plasma membrane में lipids and proteins, they are able to show a lateral movement. वो sidewise move करते हैं. So as a result of this lateral movement, what we observe is overall जो membrane हो रहा है, उसको एक fluid condition मिलता है. It is not going to be a very rigid kind of membrane. So plasma membrane का जो ये fluidity का quality आ रहा है, it's possible because of this particular quality. So that is why it is measured as its what? It is measured as its fluidity. So 109, the correct answer should be option A, fluidity. Question 110, which of the following statement is correct? This is regarding the base pairings that we observe in DNA. Adenine does not pair with thymine. ये statement गलत है. Adenine pairs with thymine. Yes, it does. Through 3 hydrogen bonds. Adenine and thymine के बीच में 2 hydrogen bonds present होंगे. So option B seems to be incorrect. Adenine pairs with thymine through 1 hydrogen bond. This is also incorrect. On the other hand, जो दूसरा base pair है, what is that? That is GC, guanine and cytosine. And guanine and cytosine, they have 3 hydrogen bonds. So guanine pairs with cytosine with 3 hydrogen bonds. ये correct है. So question 110, the correct option is option D. Question number 111, choose the correct statement. The first 2 subphases of prophase 1 are relatively short-lived as compared to pectitin. So what we remember is that prophase 1, the very first subphase is leptotin, which is followed by zygotin, and according to our question, हम इन दोनों को compare किसके साथ कर रहे हैं? We are comparing this with pectitin. तो pectitin वाला जो phase है, वो prophase 1 का सबसे important point हो रहा है. Why? Because during pectitin, we have crossing over. There is an exchange of genetic material between the non-sister chromatids of homologous chromosomes. This phenomenon is called to be crossing over, जिसमें genetic material exchange हो रहा है. So that is a very characteristic feature of pectitin. तो अगर genetic material का exchange हो रहा है between chromosomes, we need some time for that. So pectitin is comparatively a bit long. बट इन कंपैरिजन टू दैट जब हम लोग लेप्टोटिन एंड जाइगोटिन की बात कर रहे हैं, ड्यूरिंग लेप्टोटिन द फर्स्ट फेज व्हाट हैपेंस, वी हैव द ग्रेजुअल कॉन्डेंसेशन ऑफ क्रोमोसोम्स स्टार्टिंग. तो कंप्लीट कॉन्डेंसेशन अभी तक नहीं हुआ है, ग्रेजुअली इट हैज जस्ट स्टार्टेड. So it is just the beginning of the process of prophase 1. By the time it moves to zygotin में क्या हो रहा है, whatever homologous chromosomes need this crossing over to occur, these homologous chromosomes they are going to make pairs. So ये जो pairing of homologous chromosomes हो रहा है, which is also called to be synapsis, this occurs during zygotin. Again, zygotin doesn't require a lot of time. So it seems compared to the actual step of pectitin, leptotin and zygotin are comparatively short-lived. So this particular statement seems to be correct. When you cross-check, bivalent chromosomes clearly appear as tetrads during zygotin. तो जो भी paired chromosomes हैं, उनका जो tetrad वाला appearance हो रहा है, that is more pronounced in the pectitin, not in the zygotin. That statement is incorrect. Crossing over is non-enzyme mediated process. ये statement गलत है. क्योंकि अगर हमें जेनेटिक मटेरियल को एक्सचेंज कराना है, वी नीड टू रिमूव द जेनेटिक मटेरियल फ्रॉम द रिस्पेक्टिव पोजीशंस, एक्सचेंज इट एंड देन अगेन अटैच इट. सो न्यूक्लियस एंड लाइगेज एक्टिविटीज बोथ आर रिक्वायर्ड ओवर देयर. सो इट इज गोइंग टू बी एन एंजाइम मीडिएटेड प्रोसेस. ये स्टेटमेंट गलत हो गया. क्रॉसिंग ओवर इज द एक्सचेंज ऑफ सिस्टर क्रोमैटिड ऑफ होमोलोगस पेयर्स ऑफ क्रोमोसोम्स. दिस इज नॉट द करेक्ट डेफिनेशन ऑफ क्रॉसिंग ओवर. क्रॉसिंग ओवर में क्या हो रहा है, इट्स द एक्सचेंज ऑफ जेनेटिक मटेरियल बिटवीन नॉन सिस्टर क्रोमैटिड्स ऑफ होमोलोगस क्रोमोसोम्स, नॉट बिटवीन सिस्टर क्रोमैटिड्स. सो ऑप्शंस बी सी डी तीनों गलत है. सो गोइंग बाय दैट, क्वेश्चन नंबर 111, द करेक्ट आंसर इज ऑप्शन ए. क्वेश्चन नंबर 112, the prerequisite for any substance to imbibe any liquid is. This is a direct statement from NCERT, the chapter Transport in Plants. So, किसी भी imbibition के process के लिए the substance to imbibe for any liquid, what we require is इन दोनों materials के बीच में we need affinity. Affinity between what? We need affinity between the adsorbent and the liquid. जो भी material उस liquid को imbibe करने वाला है, उस adsorbent और उस liquid के बीच में we need a very strong affinity. So this is a statement directly from your NCERT. Question number 112. The correct answer should be option B. Now, question number 113. This is a diagram-based question from mineral nutrition, the nitrogen fixation part. So, if you look in this particular diagram, diagram A is representing the root hairs present in soil. So, this is before root nodule formation or any fixation. In figure B, what we have is whatever rhizobium are present in the soil, they seem to be gradually accumulating around the root hair, and they have also triggered a very important event which is the root hair curling. इसमें root hair ने एक hook जैसा structure बना लिया है. This root hair curling is important for the process of infection to occur. Figure C में हम क्या देख रहे हैं? Already root hair curl हो चुका है. That has taken up the rhizobium bacteria. Now the bacteria has entered that root hair and reached all the way up to the cortex region. तो ये जो establishment हो रहा है of the bacterial structures over there, this is the establishment of the infection thread. तो ये start हो रहा है यहाँ root hair से all the way up to the cortical cells. Now, you have to match the figure with the respective explanation. A, mature nodule, यहाँ पे तो बस शुरू हो रहा है process, no mature nodule, so not A. C, infection thread carries the bacteria to the cortex. This seems to be correct. So question number 113, the correct answer is option B. Now question number 114, the biosynthetic phase of photosynthesis requires all except. So biosynthetic phase में what we are observing is that the carbon dioxide is going to get fixed to give glucose. The fixation वाले process के लिए ATP and NADPH both are required. Carbon dioxide तो major raw material है, उसी का fixation होगा. CO2 is also required. Oxygen is not a requirement for photosynthesis. In fact, oxygen is a byproduct of the process of photosynthesis. तो किसी भी phase में हमें oxygen का यहाँ पे requirement नहीं हो रहा है. So going by that, question number 114 requires all except हमसे पूछा गया है. So what does it not require? Option D, oxygen. Now question number 115. The complete combustion of glucose in respiration is represented by, so this is nothing but the mechanism of glucose respiration. So C6H12O6, this is glucose. As glucose undergoes respiration, at the end water and energy ये दोनों formation हो रहा है. तो यहाँ पे तुम check करो the gases which are exchanged. So during the respiration of glucose, it is going to require oxygen. How many? 6 oxygen. At the end of respiration, we will see the liberation of 6 carbon dioxide. So यहाँ पे जो number A में whatever reaction is provided, that is representing the exact reaction of glucose respiration. So going by that, question number 115, the correct answer is option A. Now, question 116, match the following hormones and their function. Auxin, for example, 2,4-D can be used for what? They can be used to generate weed-free lawns. So, auxin, weed-free lawns, A4. Abscisic acid being a stress hormone, what it can trigger is, it can trigger stomatal closure. So, B3. Ethylene is important for fruit ripening. So, C5. Gibberellins or gibberellic acid, what they can trigger is, they can trigger the phenomenon of bolting in plants like cabbages. So, D2. And cytokinins were isolated from what? They were isolated from the sperm of herring fish. So, herring sperm DNA. So, E1. So, going by that, if you look at the correct code, A4, B3, C5, D2, E1. So, question number 116, the correct answer is option A. Now, question number 117, monocarpic plants are those which, so monocarpic and polycarpic, two concepts we are having. A monocarpic plant throughout its life cycle or throughout its overall lifespan undergoes flowering and fruit development only once, while polycarpic will undergo flowering and fruit development multiple times in their life cycle. So going by that, what is a monocarpic plant which flowers and develops fruits only once in its lifetime? So flowers once and after that it will die. So question number 117, the correct answer is option B. Question number 118, which of the following is a post-fertilization event in flowering plants? So flowering plants में post-fertilization के जो events हम देख रहे हैं, zygote से embryo का develop होना, then that embryo later being enclosed in a seed, being further enclosed in a fruit. These can be considered as the post-fertilization event. एक और point जो उसमें add होता है, the primary endosperm cell developing into the mature endosperm. So these can be included under post-fertilization events. Whatever development of flower, pollen grains, ovule, these are all included under pre-fertilization events. So here we have been asked regarding post-fertilization event. So embryo development. Hence question 118, the correct answer is option B. Question 119, it's a description regarding microsporangium. So microsporangium is generally surrounded by 4 wall layers. What are these 4 wall layers? Epidermis, endothecium, middle layers and tapetum. Which of the following 3 wall layers perform the function of protection and help in the dehiscence of anther to release the pollen? So outermost layer epidermis, endothecium and middle layers. इन तीनों का मिल बांट के काम ये हो रहा है that overall they will provide protection to the entire microsporangium. The endothecium, to be more precise, has a much more important role in the process of anther dehiscence. So it seems epidermis, endothecium and middle layers, ये 3 जो layers हैं, इनका protection along with that dehiscence of anther, इन सब में role हो रहा है. Tapetum has more role in providing nourishment in developing the pollen wall. On the other hand, एकदम center में जो tissue आ रहा है, that is the sporogenous tissue, and what we know about sporogenous tissue is that cells of the sporogenous tissue later mature into the pollen mother cell. Protection and dehiscence के point of view say epidermis, endothecium and middle layers, they seem to be playing a role. So epidermis, middle layer and endothecium. Option D seems to be correct. So question number 119, the correct answer is option D. Now question number 120. Dioecious plants such as papaya and date palm prevent. So what happens in dioecious is this, that the male and the female reproductive structures, and since we are talking about angiosperms, so male reproductive structure, the anthers, female reproductive structure, the carpels. So it seems that the stamens, the carpels, they are going to be present on two different plant bodies. अगर दोनों ही reproductive structures same plant body में हो रहे, then we have options of autogamy and geitonogamy occurring. But if the reproductive structures are present on two separate plant bodies, both autogamy and geitonogamy can be prevented. So in both papaya and date palm, it is observed that both autogamy and geitonogamy are prevented. Question 120, the correct option is C. Now question number 121. Among the following characters, which one was not considered by Mendel in his experiments on pea? So stem height, is it tall or dwarf? Seed color, green or yellow? Pod shape, inflated or constricted? So A, C, D. ये तीनों ही मेंडल ने यूज किए थे for his experiments. But trichomes, the nature of trichomes being glandular or non-glandular, this was not utilized by Mendel. So हमसे यहाँ पे पूछा गया है which was not considered. So question number 121, the correct answer is option B. Now question number 122, phenylketonuria is example of. So phenylketonuria, this particular Mendelian disorder, what we observe is because of the affected gene, people suffering from phenylketonuria, they tend to show a wide range of symptoms. For example, people suffering from phenylketonuria, they tend to have excessive melanin loss from their skin cells. They tend to develop premature whitening of the hairs. They also tend to develop mental retardation. तो ये जितने भी हम लोग symptoms यहाँ पे देख रहे हैं, it seems affecting one single gene seems to be controlling multiple different systems of the body or multiple different symptoms. So what idea we are getting here is that maybe जो भी original gene था, whatever was the normal version of the gene, ये मल्टीपल सिस्टम्स में मल्टीपल कैरेक्टर्स को कंट्रोल कर रहा है. तभी तो जब हमने जीन का म्यूटेशन देखा है, उन सभी सिस्टम्स में कोई डिफेक्ट आया होगा. दैट्स व्हाई वी आर सीइंग सिम्टम्स वाइड अक्रॉस द ह्यूमन बॉडी. सो गोइंग बाय दिस, इट सीम्स दिस इज अ केस ऑफ one particular gene controlling multiple characters. So what is this phenomenon called? Pleiotropy. So going by that, phenylketonuria is considered to be an example of pleiotropic conditions. So question number 122, the correct answer is option A. Now, question number 123, which of the given branch of science was developed during the period of Human Genome Project? So HGP के duration में this vast amount of data that was generated, what was required was an orderly way of storing all that data, analyzing that data, and then using these information to look into some sort of implementation. तो इतना सारा डेटा स्टोर करना, उसको अच्छे से करेक्टली फास्ट एनालाइज करना और फिर उसको इम्प्लीमेंट करना, फॉर दिस व्हाट वी रिक्वायर इज we required some new types of softwares. उन softwares का designing, उस software का application करना. So it seems for a biological system, we are now requiring new computer developments, new computer softwares. So the branch of science that was developed as a reason of this, that was called to be bioinformatics. So question number 123, the correct answer is bioinformatics, which is option B. Now question number 124, capping is defined as a process in which, so capping is a step which is included under post-transcriptional modifications in eukaryotic transcription. So in case of eukaryotes, after transcription, जो immediately RNA form होता है, that is not the mature mRNA. Instead, we call it to be the heterogeneous nuclear or hnRNA. यही hnRNA आगे modify होके results in the formation of mRNA. Post transcriptional modification में hnRNA को जब हम modify करते हैं, one thing we do is we tend to remove the non-coding regions and join the coding regions. That is called to be the process of splicing. Apart from that, इसी hnRNA का जो end portion है, जो 3' end है, उसमें हम multiple adenine repeats add करते हैं, a polyadenylate tail is added. That particular step, क्योंकि वो 3' end में हो रहा है, टेल की तरफ, दैट इज कॉल्ड टू बी टेलिंग. लेकिन यहाँ पे हमसे क्वेश्चन पूछा गया है रिगार्डिंग कैपिंग. So what is capping? Capping is considered to be the modification of this hnRNA exactly at its 5' end. So 5' end of the hnRNA, how is it modified? To that, a modified base is added, which is methylated guanosine triphosphate. तो ये जो methylated guanosine triphosphate add हो रहा है to the 5' end, ऐसा लग रहा है कि उसके head पे cap जैसा लगा दिया है. This particular process is called to be capping. So capping is defined as a process in which methyl guanosine triphosphate is added at 5' end, exactly there. So question number 124, the correct answer is option B. Now question number 125, match the following. Exons are considered to be coding sequences, whereas introns are considered to be non-coding sequences. The genetic code is based upon triplet basis on the mRNA. Whereas for DNA packaging, what we observe is the histone octamer surrounding that the DNA strand together results in a structure called to be nucleosome. So D4. So based upon this, the correct code is A1, B2, C3, D4. 1, 2, 3, 4. So correct answer is option C. Now question number 126. Thalassemia and sickle cell anemia are caused due to problems in globin molecule synthesis. Select the correct statement. तो जितने भी यहाँ पे statements आ रहे हैं, they seem to be describing the conditions of thalassemia and sickle cell anemia. So what we recall is sickle cell anemia. In this case, what happens? Globin synthesis बंद नहीं हो जाता है. But जो भी globin particle यहाँ पे बन रहा है, whatever globin molecules are formed, it seems these globin molecules are a bit deformed. वो गलत बन रहा है. So it seems if the quality of the globin is affected, that will further affect the quality of the RBCs. That is why in sickle cell anemia, instead of having the normal biconcave shape of RBCs, RBCs appear to be sickle shaped. तो यहाँ पे क्या हो रहा है? In sickle cell anemia, the quality of the globin or the quality of the RBC is affected. So we can call sickle cell anemia to a qualitative disorder of blood of RBCs, or let's say globin. On the other hand, when we study thalassemia, in thalassemia what happens is there is going to be a decrease in one type of globin. So we have alpha and beta globin coming together to make the complete globin molecule. So what we observe is अगर alpha globin का amount कम हो गया तो उसका respective जो beta globin है that seems to be more in amount now. क्योंकि जो भी अल्फा एंड बीटा ग्लोबिन बन रहा था, they were supposed to come together. लेकिन अगर अल्फा ग्लोबिन का अमाउंट कम हो गया, respectively what will happen? The beta globin amount seems to have been increasing. So all these beta globin molecules, they will start to accumulate in the RBC. Continuous accumulation of the other type of globin that will result in eventually cell lysis. So what is being affected here? एक टाइप के ग्लोबिन का क्वांटिटी कम हुआ, जिसकी वजह से इवेंचुअली आरबीसी का लाइसिस हो गया. सो हेंस रिजल्टिंग इन द डिक्रीज इन द क्वांटिटी ऑफ द आरबीसीज. हेंस वी कैन से दैट थैलेसीमिया कैन बी कंसिडर्ड टू बी अ quantitative disorder of blood. So in both globin is affected, but SCA is qualitative whereas thalassemia is quantitative. So going by that, thalassemia is a quantitative defect in globin while SCA is qualitative. This statement is correct. So question number 126, the correct answer is option C. Now question number 127, you have to pick out numbers 1 and 2 who are included as steps of classical plant breeding. So after collection of germplasm, the next step is going to be to select which parent is used for cross hybridization, because next step ही क्या हो रहा है, cross breeding या फिर hybridization. So यहाँ पे कौन सा step आना चाहिए, selection of parents. Selection of parents. Next, एक बार hybridization हो गया, and before you can release and commercialize these cultivars, what you need to do is, after hybridization, whatever recombinants you are getting, you're supposed to evaluate and test those recombinants. That are these up to the mark or not? Do they contain whatever desirable qualities were needed? So going by that, number 2, what it should be? Testing of superior recombinants. So going by that, question number 127, the correct answer is option A. Now question number 128. During nitrogen fixation, reduction of one molecule of nitrogen into two molecules of ammonia consumes how many molecules of ATP? So for two molecules of ammonia to be produced from one nitrogen, we require 16 molecules of ATP. Hence question number 128, the correct answer is option B. Now question number 129, this is the direct statement from NCERT regarding the findings of Human Genome Project. So what Human Genome Project told us that an average gene consists of 3000 bases, but their sizes vary greatly, with the largest known human gene being dystrophin, so dystrophin with 2.4 million bases. So 3000, dystrophin, 2.4 million bases. Question number 129, the correct answer is option A. Now question number 130, select the incorrect statement about prokaryotic ribosomes. So for prokaryotic ribosomes, incorrect statement. 50S and 30S subunits unite to form 70S ribosomes. This statement is correct. Ribosomes are the site of protein synthesis. Again, directly a correct statement. Polysomes indicate synthesis of identical polypeptides in multiple copies. If you see, option C and D both are related with the concept of polysome. So when we study polysome or polyribosome, what we mean by this is, in polyribosome, with one mRNA molecule, what we have is the association of multiple ribosomes at the same time. So it means one after another to one mRNA, एक के बाद एक क्या हो रहा है, multiple ribosomes attach हो रहा है. So what can happen as a result of this is, in that one mRNA, अगर सिर्फ एक ribosome आके attached होता, so after it completes reading the mRNA, I'll just get one copy of the polypeptide. But एक के बाद एक अगर multiple वहाँ पे ribosomes attach हो रहे हैं, so as each ribosome is detaching, I'm getting one copy of polypeptide, the second one removed, the second copy of the polypeptide. So as a result of which, I can get multiple copies of polypeptide from that same mRNA, same protein के या same polypeptide के मुझे बहुत कम time पे multiple copies मिल रहा है. So it seems polysomes are a very efficient way of having protein synthesis. So going by that, statement C seems to be correct, that यहाँ पे synthesis of identical polypeptides in multiple copies is actually happening. So statement B, polysome consists of many ribosomes attached to tRNA. ये वाला point गलत हो गया. So in polysome, the attachment is going to be happening with one mRNA, not tRNA. So हमसे incorrect पूछा है. So question 130, the answer is option D. Now question number 131. In the absence of an external source of water, kangaroo rat in North American desert is capable of meeting all its water requirements through. So what the kangaroo rat contains is, it contains an advanced mechanism of internal fat oxidation. As a result of this internal fat oxidation, it can generate metabolic water, which can compensate for whatever water scarcity it is facing in the desert condition. So going by that, question number 131, the correct answer is option A. Question number 132, you have been given 3 pyramids respectively to age pyramids. So select the correct option with respect to age pyramid. तो number A, यहाँ पे क्या देखने को हमें मिल रहा है, that compared to pre-reproductive, the amount of reproductive people are less. तो इस pyramid का base बड़ा हो रहा है. As a result of which what will happen, this particular population will eventually keep on growing bigger. तो ये क्या हो रहा है, एक बहुत steady rate में ये population extend होता जा रहा है. So this appears to be the pyramid of an expanding population. Whereas pyramid B में क्या हो रहा है, pre and post reproductive, इनको अगर हम compare करें with respect to reproductive, almost एक similar line पे ही आ रहा है. More importantly, the pre-reproductive and reproductive, ये दोनों तो almost same level पे आ गए हैं. तो इस वजह से ये जो particular population है, over the course of the next few years, इसमें कोई बहुत ज़्यादा difference नहीं आएगा. So because of this, it seems that the particular population here appears to be stable in nature. So this is the pyramid for a stable population. लेकिन last pyramid में क्या हो रहा है? When you look at this, the number of reproductive seems to be more than pre-reproductive. So as a result of which what happens, जब pyramid का ये base यहाँ पे छोटा हो गया है, so pre-reproductive में कम individuals होने की वजह से यहाँ पे what will happen is in the next few years this particular population it will start to decline. So this is the pyramid for a declining population. So expanding, stable, declining. Going by that, question number 132, the correct answer is option A. Now question number 133, the primary productivity depends upon. So primary productivity किसी भी area में dependent होगा mostly upon their photosynthetic organisms. So what we are observing here is that photosynthetic capacity of plants that is going to decide primary productivity. But along with that, अगर plants को अच्छे से photosynthesis करना है, so they are going to require what? Minerals. So what we observe is in aquatic ecosystems, primary productivity tends to be a bit limited. Why? क्योंकि उन aquatic ecosystems में plant तो बहुत ज़्यादा amount में हो सकता है. Even if we do not have higher plants, you can still have other photosynthetic organisms. लेकिन इतने photosynthetic organisms होने के बाद भी aquatic ecosystems में productivity comparatively terrestrial से क्यों कम हो गया है? Because water में जो nutrient availability है, whatever nutrients should be given to the organism, वो amount में थोड़ा कम होता है. So it seems photosynthetic efficiency में कौन सा nutrient कितने amount में अच्छे से available है या नहीं है, this will also play a role. So अगर दोनों points को हम bring together करें, so availability of nutrients and photosynthetic capacity of plants both have a role to play in deciding the primary productivity of an area. So going by that, both A and B are correct. So question number 133, the correct answer is option C. Now question number 134, the functional components of ecosystems are, ecosystem में 4 functional components हो रहे हैं. Productivity, that's one functional component. Along with that, decomposition, nutrient cycling and energy flow. Succession and stratification, they are not considered to be functional component of the ecosystem. So going by that, A, B, C, D, these are the functional components. So question number 134, the correct answer is option B. Now question number 135, which one is not an example of alien species? So Parthenium, commonly known as carrot grass, this was introduced as a contaminant in India along with imported wheat seeds. And after that, Parthenium spread across the Indian subcontinent and it is a very problematic invasive or alien species. So Parthenium is an example of alien species. Acornia, commonly known as water hyacinth. Again, water hyacinth is not naturally found in India. They introduced water hyacinth in the aquatic ecosystems because of its beautiful leaves and flowers. But again, what was observed was Acornia can very rapidly grow in water bodies, eventually suffocating the living aquatic ecosystem. So again, Acornia is an invasive species. Clarias, this is the African catfish which was introduced again in the aquatic ecosystem of India. The Clarias, this African catfish, it eventually resulted in the decline in the population of the native catfish species. So this is also an alien species. Triticum, on the other hand, that is the genus to which the common wheat belongs to. And wheat, that is an important cereal crop that we have. So that is not an alien species. So alien species कौन सा नहीं है हमसे पूछा गया है. So question 135, the correct answer is option B. Now moving on to section B. Question number 136, which of the following bacteria are responsible for the production of biogas from the dung of cows and buffaloes? So biogas का एक major constituent हो रहा है methane. So methane production के लिए जो bacteria यहाँ पे involve हो रहा है, that is methanogenic bacteria or methanogens. So going by that, 136, the correct answer is option C. 137, in which algae sexual reproduction is oogamous and accompanied by complex post-fertilization development? ये जो पूरा statement है that sexual reproduction is oogamous and it is followed by complex post-fertilization development. This is a characteristic feature of rhodophyceae or red algae. Now all we have to do is, out of the 4 options, we need to find out which is the member of red algae. So Porphyra, option D, that is a member of red algae. So question 137, the correct answer is option D. Now question number 138, thorn of citrus and bougainvillea is modified So citrus and bougainvillea plants में that sharp pointed structure that they develop as a defense mechanism, that is called to be the thorn. And thorn का development कहाँ से हो रहा है? Thorn is developing from the axillary bud. So axillary bud, that is a part of the stem. So overall what we can say is that the thorn is considered to be a stem modification. So 138, the correct answer is option A. Question number 139. When xylem and phloem are arranged in an alternate manner on separate radii, such a vascular bundle is called. तो ये जो particular statement आ रहा है on separate radii, that itself will give us an idea that the arrangement of xylem and phloem on separate radii, that is actually considered to be a radial vascular bundle, which is a characteristic property of roots. Let it be monocot or dicot roots. So question number 139, the correct answer is option A. Now question number 140, which of the following statements is correct? The core of cilium or flagellum is the basal body. This statement is incorrect because cilia and flagella का जो core है, that is called to be axoneme. Elaioplasts store starch, whereas aleuroplasts store proteins. Now, aleuroplasts, they do store proteins, but elaioplasts are not storing starch. Elaioplasts are storing oils, fats, lipids. The starch storing plastid is amyloplast. तो ये statement भी गलत है. Statement C, membranous extensions into the cytoplasm in cyanobacteria which contain pigments are called chromatophores. This particular statement is correct. Chroma means color. So chromatophores are those membranous extensions, specifically in case of cyanobacteria, which are going to develop pigments, जहाँ पे photosynthesis occur होगा. So question number 140, the correct answer is option C. Now question number 141, the moiety present at the 5' end of ribose sugar in a polynucleotide is. So let it be a polynucleotide consisting of any particular nitrogenous base. That 5' end that we have, what exactly is it going to contain is, it is going to contain a phosphate group. So 141, the correct answer is option C, phosphate. Now question number 142, which of the following phases lasts for more than 95% of the duration of cell cycle? So in the entire cell cycle, it can be divided into interphase and M phase. M phase is the phase where we actually have karyokinesis and cytokinesis occurring. लेकिन उसके लिए जो preparation हो रहा है, interphase, including G1S and G2 phase. So this preparatory phase that we have, that is actually taking up a very large amount of time of the total cell cycle. So almost 90% और 95% से भी ज़्यादा का जो time है of a cell cycle वो कौन cover कर रहा है? That is covered by the interphase. So question number 142, the correct answer is option D, interphase. Now question number 143, the root nodules formed during biological nitrogen fixation. So inside the root nodules what we have is the bacteria are going to be present, rhizobium, who are going to fix atmospheric nitrogen into ammonia. Now ये जो ammonia उसने fix किया, this ammonia needs to be taken up by the plant and utilized. क्योंकि वो already plant body के अंदर present है, what the rhizobium will need to do is, it will need to make sure that the ammonia is generated in such a manner कि वो already जाके plant के vascular system में enter कर चुका है so that the plant can transport it. On the other hand, as a result of this symbiotic association, what is the rhizobium getting in return? The rhizobium is getting nourishment, it's getting food from the plant. So again उसके लिए भी क्या चाहिए? It should be connected with the vascular tissues, it should be connected with the phloem so that it can directly get the food from there. So it seems when a root nodule is established, we need a very close association with the plant's vascular system. So if you keep this point in mind that root nodules formed during biological nitrogen fixation, it establishes a direct vascular connection with the host for exchange of nutrients. So statement B is correct here. So question number 143, the correct answer is option B. Now question number 144. The breakdown of proton gradient results in the formation of. So the breakdown of proton gradient, this occurs during both photosynthesis and respiration. तो ये जो particular statement है, ये correlate कर रहा है with the chemiosmotic hypothesis. In chemiosmotic hypothesis, we see that across a suitable membrane, a proton gradient is maintained. After some time, उसी membrane के through, what we will see is the breakdown of this proton gradient, and along the way, it is going to utilize an F1 F0 particle in order to synthesize ATP. This hypothesis holds true for photosynthesis. This hypothesis also holds true for respiration. So it seems proton gradient का breakdown required है किस काम के लिए? To generate ATP. So question number 144. The correct answer is option D. Now question number 145. The formation of acetyl coenzyme A from pyruvic acid is the result of its. So when pyruvic acid undergoes an event of oxidative decarboxylation, where it's undergoing oxidation, simultaneously उसमें से carbon dioxide भी remove हो रहा है. So this oxidative decarboxylation of pyruvic acid results in the formation of acetyl coenzyme A. Hence, question number 145, the correct answer is option D, oxidative decarboxylation. Now, question number 146. Increased vacuolation, so more formation or more pronounced formation of vacuoles, cell enlargement, the cell growing in size, and new cell wall deposition, so primary में अभी secondary cell wall भी add हो रहा है, are the characteristic of cells in phase of growth. So which particular phase of growth is this? This is going to be the phase of elongation. So इसके पहले phase of meristematic activity में cells were just actively dividing. इसके बाद आएगा phase of maturation or differentiation, where the cells are going to acquire a permanent structure and function. But intermediate, जिस point पे cells का division बंद हो रहा है, and then they increase the number of vacuoles, they tend to increase the size, add new cell wall depositions, that comes under the phase of elongation. So question number 146, the correct answer is option B. Now question number 147. Filiform apparatus is the characteristic feature of. So what we remember about a mature embryo sac is this, that let's say this is the chalazial end, this is the micropylar end. So chalazial end may we have the presence of 3 cells. These are called to be the antipodals. Now very center we have the presence of the central cell, and inside this central cell we have the presence of the 2 polar nuclei. Micropylar end की तरफ we will have the presence of the female gamete, the egg cell. Now what we notice is micropylar end की तरफ we have the presence of some finger-like projection, and from these finger-like projection we have the extension of the synergid. So this is the whole arrangement of a mature embryo sac. तो ये जो finger-like projection micropylar end की तरफ आ रहा है, this is called to be the filiform apparatus. So it seems that the filiform apparatus is associated with what? Synergids. So going by that, question number 147, the correct answer is option A. Now question number 148, this is a numerical from Principles of Inheritance and Variation. A female whose father was colorblind. So first you look at the parents, the female parent and the male parent. Before we read the question further, let's assume they are normal. Normal in the sense that they are not carrying any mutated genes or anything. हम normal male and female का genotype यहाँ पे note किया है. Now based upon the information of the question, we are going to look at are they really going to carry any mutation or not. So for the female, what is mentioned, a female whose father was colorblind. So this particular female, whatever father she had, that father was colorblind. C over here is denoting the mutated gene for colorblindness. So if her father was colorblind, so definitely one of her X chromosomes were inherited from that colorblind father. It means वो XC वाला जो mutation है, she must have been carrying. So what that tells us is that this particular female, she is unaffected, but she is a carrier. Now this male marries a normal male whose father was also colorblind. So this particular person, his father was also colorblind. But this is not going to affect this male because अपने father से इस male ने सिर्फ वो Y chromosome inherit किया है, not the X chromosome. So this male is normal. So now we have what? Now we have the genotypes of the 2 participating parent. Yes. So now we're going to look at the gametes. Female gametes. half of them carrying normal X chromosome, the other half carrying mutated. Male gamete दोनों halves ही normal है. Now when you combine the presence of unaffected females with no mutation, the presence of unaffected females but carriers, the presence of completely unaffected males and the presence of some affected males. So what we can see is that out of the total son, 50% son are unaffected, 50% son are affected. Out of the female or out of the daughter, all the daughters are going to be unaffected. It's just that 50% without any mutation, 50% being carriers. So अब question में हमसे पूछा गया है, what is the probability that their daughter will be colorblind? Any of these daughters you pick up, none of them are ever going to be colorblind in nature. So the probability of having colorblind daughter is going to be what? 0. So question number 148, the correct answer is option A. 0% probability of having a colorblind daughter. Now question number 149. DNA and RNA differ from each other with respect to. In DNA and RNA जो phosphate group है that is common. Phosphate में कोई difference नहीं आ रहा है. पहला major difference आता है DNA deoxyribonucleic acid contains deoxyribose sugar, whereas ribonucleic acid RNA contains ribose sugar. So sugar का difference आ रहा है. The second difference that we have are the nitrogen bases. दोनों में ही purines pyrimidines हो रहे हैं, but how exactly are the purines and pyrimidines present? In DNA we have adenine, thymine, guanine, cytosine, whereas in RNA we have adenine, uracil, guanine and cytosine. So thymine uracil वाला difference आ रहा है. So in DNA and RNA the sugar and the nitrogen bases would be a point of difference. So nitrogen bases and sugars. Question number 149. The correct answer is option D. Question number 150. The term the evil quartet is related with. So evil quartet, there should be 4 things here. So what are the constituents of the evil quartet? Habitat loss and fragmentation, overexploitation, co-extinction and alien species invasion. So ये जो 4 points are exactly what are they explaining? These are considered to be the reasons behind biodiversity loss. So these 4 together are considered to be the evil quartet, the 4 major reasons behind biodiversity loss. So the term evil quartet is related with 4 major causes of biodiversity loss. Question number 150. The correct answer is option D. So I hope you understood the discussions today. Make sure you do not repeat the same mistakes. Continue revising and good luck for your upcoming